speaker. It's a very great pleasure to introduce Sarah Teichmann. Sarah Teichmann did her degree in biochemistry in Cambridge and then went on to do a PhD under Cyrus Chothia at the MRC laboratory in Cambridge. She did a one-year fellowship in Janet Thornton's lab before becoming a program leader at the MRC laboratory. And she was also made a research fellow of Trinity College at the same time. She stayed there for till 2013, when she became a group leader, a joint between EBI and Sanger, and also a research associate of the Cavendish lab, which is in the physics department at Cambridge. And she was made director of the Cavendish lab in 2015. And in fact, this year, she has been given the very prestigious position of head of cellular genetics at the Sanger Institute. So Sarah had an early interest in proteins, I think. She did an undergraduate project in NMR with Enes Lawi before moving to Cyrus Chothia's lab. And there she did an analysis of all the emerging genome data in the late 90s using structural domain annotations from SCOP and looking at the distribution of domains in genomes. Significant proportion of proteins, she discovered that a significant proportion of proteins had multi-domains. And she did, I think, a very elegant and beautiful analysis of the mosaic evolution of proteins. Then, returning somewhat to her structural roots and building on new ideas in networks, she used graph theory to analyze the assembly and the emergence of complexes. And as with her genome analyses, I think she did very pioneering work in this area and developed a very well-established world-leading resource in protein complexes, 3D complex, and actually a periodic table of complexes, which I personally think is really neat. More recently, she's expanded her research interests even further to include transcription factors and global regulation of gene expression. She's improved the annotation of transcription factors in genomes and used that to analyze transcription factor networks in yeast and E. coli, more recently using RNA-seq data and the powerful tool of single-cell transcriptomics to really dissect out the noise and examine cell-to-cell variation in transcription factor networks. She has very many prizes, too many to mention, I think, but just a few. She got the Max Perutz Prize for her PhD. She was made an EMBO member in 2012, and she got the gold medal, the EMBO gold medal, in 2015. She has a Lister Prize. She has the Colworth Medal for Biochemistry. She's a Fellow of the Academy of Medical Sciences in 2015, and this year a Fellow of ISCB. Just a couple of quirky things. In 2009, she was made the Shell Woman of the Future, and she's also been coordinating a project in which a piece of music was composed for a string quartet, a piece of music called Hearing Your Genes Evolve. And on that note, I just say we look forward to hearing your talk. Thank you very much.
Thank you so much, Christine, for your kind words. And thank you, Christine and Teresa and Alfonso and Bonnie and others for inviting me to speak here and for the honor of becoming an ISCB Fellow. I should say it's really special to be recognized by this community because my roots are in computational biology, and exactly 20 years ago, I actually attended a course which was very ISMB, ISCB-like, where Burkhard was a teacher. So this is one of the first conferences during my PhD, and ISMB was also one of the first conferences, the one in Heidelberg, that I went to during my PhD. So this is extremely exciting, and it's a great honor for me to be here and to be able to speak to you. I've been to SIGs before, which are of course sort of legendary, but I haven't spoken in the main ISMB, so this is really nice. And what I want to share with you today is our journey that we've been going on for the past 5 years or so through the wonderful world of single-cell genomics. And what you can see on this slide is fluorescence microscopy images of individual cells with reporter proteins or fluorescent antibodies, where you can see that in samples of these different cells, the fluorescence, reflecting the protein abundance, is varying between the individual cells. So clearly, even in a sample that you expect to be a fairly homogeneous cell line or set of cells, there's variation. And the question is that variation interesting? And I really stumbled across this in the days when we're using bulk RNA sequencing, looking at T cells, which is one of our favorite cell types in my group. And we sort of delved into the world of single-cell gene expression using single-molecule RNA FISH for a couple of factors, GATA3 and TBX21. These are two key transcription factors that specify the cell fate determination between two different lineages of T cells. And whereas with bulk RNA sequencing, you of course see a single expression level from your cell sample, this collaboration with Alexander van Oudenaarde, where we use single molecule FISH, really opened my eyes to the fact that you have this broad distribution of the master regulator GATA3 in our cell sample here, which is 500 cells. And you can see that the number of cells is on the y-axis and the number of transcript in each cell is on the x-axis. And the number of mRNAs per cell is varying from 1 to almost 350. So there's a broad distribution of this master regulator, which is key for these Th2 cells. And so my expectation was that it would be very tightly regulated and there'd be the same number of transcripts in each cell, but you can see that that's not the case. And this isn't just because of cell proliferation in the cell cycle, because although the cells are growing, you'd expect at most a twofold variation. You can see that there's lots of noise here. And the same thing is true for TBX21, which is a transcription factor that's thought to be suppressed by GATA3. And yet you can see that's true in about 400 of the cells where there's 0 transcripts. And this is after noise filtering for the technical noise in the microscopy. There are still about 100 cells, so 20% of the population, where there's between 1 and 40 transcripts per cell. So this transcription factor that's actually a master regulator for a different lineage is kind of popping up stochastically in these cells. And that made me realize what we really want to get to, and this is sort of more than 5 years ago when we were doing this, 7 years ago or so. What we really want to get to is to be able to sequence the transcriptome of every single cell. And then we'd be able to understand the heterogeneity in these very plastic T cell lineage and in many other cells in mammalian tissues. And so with single cell RNA sequencing, if you have a sample of cells, a cell population that's heterogeneous like this, then with the bulk RNA sequencing, as I said, you get the mean expression value, the weighted average of the expression of that gene in the different subpopulations of cells in your sample. And so you lose any insight into the cell population diversity because of this averaging. With single-cell RNA sequencing, and this has been introduced, I should say, very nicely in the keynote this morning and by other talks in the conference, I just want to sort of recap. What you get for free, sort of, by unbiased sort of data mining is how many cell types there are, and we heard about the clustering this morning. You get the frequency of each cell type, and the beauty of it is that it's unbiased, because you get the whole transcriptome in each single cell. So it's unlike that single molecule RNA FISH experiment that I was showing you, or unlike FACS, where we have to ask for protein markers and antibodies. And so you can think of this as sort of an exercise in extracting order from a bioinformatics point of view from heterogeneity. So if we have a population of cells like this crowd of people in Times Square on New Year's Eve, then what we want to do is basically align, let's say, the cells in some sort of pseudotime. So you could think about aligning, taking all the people and ordering them by age, and you get some sort of average picture of the life cycle of a human being if you had all the young, middle-aged, old, and so on aligned. You could think about clustering the people, so Americans of European origin, Americans of African origin, and so on, and you get some average picture of the features of those people. And so what we've had lots of fun with is grappling with these computational problems over the last few years, and I'll talk a little bit about that as we go through the talk, about the different methods that we used. And I'll introduce an assessment of the sensitivity and specificity of different single-cell RNA-seq protocols, because that's there's something, although I'm driven by the interesting biological questions there's something somewhat quantitative inside me that wants to know exactly what we're measuring. And we've used spike-ins to assess this. And the second two parts, I'll be telling you two different biological stories and illustrating the computational methods that we use throughout those analyses, one on mouse embryonic stem cells and one on T-cell clones and cellular decision-making. So when we started off, the main method that we used was chip microfluidics for cell capture on the fluid MC1. You can see these little wafers. We now use many different capture methods, but this was a real eye-opener because we were having a lot of trouble in those days, as like 6-7 years ago, with FACS sorting individual cells. And this chip allowed us to capture these little yellow balls, which are the cells, in the chambers. And within half an hour, 96 cells are captured, and you can take the chip out, inspect it microscopically, see whether there's 0, 1, or 2 cells, and put the chip back in as quickly as possible to avoid stressing or killing the cells, and then the robot goes off and lyses the cells, which you see here. The RNA pops out, as does the DNA, of course, but we're focusing on the RNA in this talk. And then reverse transcription happens automatically inside this, in nanoliter volumes, basically, inside these chambers. And thermal cycling leads to amplification of the cDNA then, because there's thermal control on the chip. And then you can take off 96 for SmartSeq, or up to 800 cells in the FluidAm system now for ScrubSeq. Do barcoding and library prep and so on, and send for next generation sequencing, so you have your transcriptomes. The data comes back, and the fun begins. And as I said, when we were first doing this, one thing that I was sort of obsessed by was knowing whether what we're measuring is just a function of the technical variation between cells, the technical variation between the chambers, or whether this is true biological variation. And obviously that's critically important to any downstream analyses and biological conclusions that you draw. And so this was just during the time that I was moving from, Christine mentioned that I moved from the MRC Laboratory of Molecular Biology in the center of Cambridge, or on the southern edge, out to the Genome Campus in Hingston. And that was a very exciting time, and there was lots of stuff kind of happening because we were moving, I was having a baby, and so on and so forth. And what was really important to me, though, was to know what the technical noise was. And during this experiment by Ola Kolodzic and Valentina Prosorpio in my group, I made really sure that they spiked in the Ambion ERCC92 kit, which are these red dots here. And during this move to the Genome Campus, I bumped into John Marioni, a statistician, and that was a really lucky move, because he's my office neighbor in EBI, and he linked us up with Marcus Heisler's group in Embl Heidelberg, who were actually thinking about similar things, but in plant cells, and they were using commercially available human RNA spike-in. So analogous computational or sort of quantitative problem, different system. And John and Simon Anders from Wolfgang Huber's group then developed this nice statistical framework with John Kim, where cell-to-cell variation is plotted on the y-axis as the coefficient of variation squared, so basically the width of that distribution, the standard deviation divided by the mean squared, and gene expression here, normalized counts on a log scale, are on the x-axis. And what you can see here is that the spike-ins are fit by a quadratic, and these are the big red dots. And so what it means is that basically the noise is increasing with a lower concentration of spike-ins. So these are high abundance spike-ins, these are low abundance spike-ins, and basically the noise is dominated by abundance or expression level. That's not that great news on the one hand. On the other hand, if we have a confidence interval, we can segment out the genes that are varying beyond the technical noise. And this is about 900 mouse genes, these purple genes here. And very reassuringly, those 900 genes were related to cell proliferation, so the cell cycle. And as I mentioned, these T cells are proliferating in the cell culture differentiation, the experiment that we did here. and also to T-cell differentiation. This was a naive to Th2 cell culture. And so we can expand this principle now. And what Valentin Svensson, my group, has done, and this is unpublished work, is basically suck in all the published data sets that have spike-ins, and many of them actually use this kit. It was first used by Itai Yanai in CellSeq in his Cell Reports paper. And then compare the sensitivity and specificity of the different data sets using different protocols. So basically what we're doing is taking data sets on different, which may have mouse or human or various different cell types, but we're ignoring the mammalian genomic reads and we're focusing on the spike-in mix here to compare the performance of the different protocols. And we calculate the lower detection limit. Valentin did this with a logistic regression. And that allows him to estimate the minimum number of molecules required in the cell or in the sample, basically in each individual sample within the batch, to be detected at the end of the day by the next-gen sequencing. And what you can see here is that the lower detection limit varies quite a lot between the different protocols. So I mentioned CellSeq, and that has a lower detection limit of about 100, and then MarsSeq sort of varies 100 to 1,000 in this dendritic cell paper. The smarter, which is the only full-length transcript protocol, and that's going to become important later. So these are both 3' tag protocols that only get the 3' end of the transcript. These are this one is performing as the most sensitive, and then there are many others that I haven't fit on this slide that we've got now. So the sensitivity is quite different, but the good news is that the accuracy in terms of quantifying gene expression measured as the correlation coefficient between those spike-in abundance levels that we know the concentration of when they're put in, and we know how many mRNAs there should be there relatively between the 92 spike-ins. We can then measure their relative expression levels in TPM and calculate the correlation coefficient as a measure of accuracy. You can see that the accuracy is actually sort of at least 0.7 sort of, and it goes up to almost sort of over 0.9 for all of the protocols. And this was quite impressive to me, because it means that in each single cell, and across the batch, we're quantifying gene expression fairly in a reasonably accurate way. Now this does, if we focus on low input molecules, then the accuracy is a bit less. So the quantification is more reliable for higher abundance genes. That won't be too surprising to you. And the mean lower detection limit is anti-correlated with the accuracy. So the more accurate a method is, in general, the more sensitive it is as well, and particularly for these lowly abundant, lowly expressed species of molecules. Now, these tails that you see here coming from each batch are subsample data, and so what that means is that we can simulate differences in sequencing depth. And what we wanted to know was, how deeply do we need to sequence? How much money do we need to spend in order to get a particular level of information? And what you can see here is that if we do this subsampling experiment, then we get an increase we get a benefit in terms of a better lower detection limit from sort of 10 to the 5 of 100,000 to 1 million reads, but then you saturate and there's no more benefit after about 1 million reads. So that's telling us basically, we're just throwing away money if we're sequencing more than 1 million reads per cell. And we can probably get away with a lot less, depending on our research question, kind of that we're asking of the cells. So this is just to say that, this is using these spike-ins as a way of assessing sensitivity of the protocols. We want to know what we're dealing with, and we see a wide range of sensitivities over several orders of, more than an order of magnitude. We can calculate that there is a sequencing benefit in increasing sensitivity up to about 1 million reads. And overall, all of the protocols have pretty good specificities. And so armed with that information, we can then proceed to apply, whichever method is most suitable to our biological questions. And I want to thank Valentin Svensson, and also who spearheaded this analysis, and has been working on this for a long time, and part of that is because we're actually generating our own data, kind of to make sure that this analysis is stable when you use in the same hands, with the same cells, with the same spike-ins, and so on. So this is work in progress, but we think that basically the conclusions hold. Lamha Lee isn't on here, but she's an intern in the group who's contributed to this reanalysis of this data. And also thanks to Anna Tveit and Ian Macaulay, who donated different protocols to us before they were published. So in summary, now knowing that these single-cell RNA-seq protocols are doing something sensible, we can go ahead and we can ask, let's test drive this method now and see whether we can detect subpopulations in mouse ES cell cultures. Okay, why is this interesting? It's basically because stem cells, and when I was moving to Hingston, there were ES cells everywhere because there's a large mouse knockout program there. So there's a lot of interest in the optimal embryonic stem cell culture. And the sort of traditional culture is serum LIF, which is serum with leukemia inhibitory factor or growth factor. And when the stem cells are cultured under this condition, then they actually look heterogeneous. So some are larger, some are smaller, they're different in shape and size, and it's known that there are nanog fluctuations across the cells. Now, Austin Smith had a really good innovation quite a few years ago now, where he added 2 inhibitors to the culture, and these 2 drugs stabilize the culture, and indeed the cells actually look more homogeneous under the microscope, and nanog is expressed at more stable levels across the cells, that's known. And then more recently there's a Japanese other culture which we call alternative 2I, which uses 2 other drugs. And so what we wanted to ask was, if we profile the same cell line in the 3 different cell culture conditions, then we should be able to recapitulate those morphological differences in cell-to-cell variation using the genomics, using the single-cell transcriptomics. And so Ola Kolodziejczyk, who's a really talented Polish PhD student in the group, went away and did these experiments with help from Pental Lu's group and Jason Tsang. And you can see here, Ola's cells are over here. So the green is the serum LIF cells. And you can see these are spread apart, and there are 2 subpopulations that I'll tell you more about later. The orange cells are the alternative 2I, and these blue cells with a little purple subpopulation are Austin Smith's 2I, 2 inhibitor culture. And then we're superimposing these cells onto the embryonic time course from Richard Sandberg, the SMART-seq data on mouse embryonic development. So this is all the way from sort of 2-cell stage 4, 8, 16, 32, 64 to the blastocyst stage down here. It's a different strain of mouse from what we used, but the genetic differences doesn't seem to make we can fit this developmental time course, and you can see that the proximity of the blastocyst cells from which the embryonic stem cells are made is really close to the 2I cells on this 3D PCA, at least as a measure of distance. And so what that's telling us is that the 2I stem cells are mimicking the blastocyst stage and the pluripotent stage most closely in terms of their overall transcriptomes. And there are some cells over here, these little purple ones, that are positioned even more closely. And it turned out, and this took some filtering or some digging to get this out, these are so-called 2C cells that have been described in the literature by McFarlane et al., which have, interestingly, an endogenous retrovirus expressed in them, and they're about, I think, 5 or a couple of % of the cell population. And the interesting story here is that these cells actually didn't cross our quality control filter. So we use mappability, mitochondrial read, and a bunch of other features in our Cellity pipeline that we published in Genome Biology recently to filter out cells that are garbage, that are apoptotic, broken, debris, and so on and so forth. And these cells didn't actually cross our quality control filter, and it turned out in the end that they were, that was because they had a lot of reads from an endogenous retrovirus that was popping up and not, these weren't mapping to the mouse genome. But we rescued them back once we'd realized that. And you can see here, these are actually very close to the blastocyst. They're not close to the 2-cell stage of the embryo, so we don't think that they're really mimicking that stage. but they are very close to the blastocyst stage. And then you can see here the alternative 2I condition is a bit further away, serum LIF is a bit further away, and you have the diverse subpopulations that I'll go into later. So those are the embryonic stem cells. And I'll just say, if you remember the original question that we're asking, what we want to see is there a difference in the global noise between these serum LIF cells that we know are morphologically heterogeneous and the 2I and alternative 2I cells where the drugs are thought to make the populations more homogeneous with homogeneous nanog expression and so on. And so in order to address that question, what we need is a measure of cell-to-cell variation in gene expression. And what you can see here, the coefficient of variation squared that we used here is clearly not suitable because it's very dependent on expression level. So it's dominated by expression level. And so what Kim did with John was to adapt this and regress out the noise, and that's essentially what's shown here, is that we use we can use a windowing approach or a spline and basically develop the distance to the median of the coefficient of variation squared to calculate the noise of each gene in a cell sample, in a cell population. And if we plot this dm, or distance to the median of the coefficient of variation, we can see that actually the global distribution of that noise measure is exactly the same in all of the culture conditions. And these are two different batches of serum, just to show that. We obviously regressed out batch effects and so on, and can recapitulate the same distribution in the two batches. And so this was really surprising because, as I said, we know that there are nanog fluctuations in serum, but not in 2I. So we know that the cells look large, small, different shapes and sizes in the serum, but not in 2I. So why are we getting the same global noise across the conditions? So in order to dig down to the bottom of this, what Kim did was to group the genes into bunches, the gene ontology categories, and plot the noise of different gene ontology categories using DM in serum relative to 2I and 2I relative to serum. And that's what you see on this axis, the fold change in noise for a functional category. And on the y-axis is the expression level. And one gene ontology category, one functional category that stood out as much more noisy in 2I, which is the one that we expect to be homogeneous with homogeneous nanog expression and so on, is mitosis. So in other words, the cell cycle. That actually surprised us, but we sort of delved in a bit more. We extracted genes that cycle throughout as the cells proliferate from cycle base. And you can see these are G2M genes here in the rows, and the cells are in the columns. And the interesting thing is that these G2M genes are highly expressed in some 2I cells and lowly expressed in other 2I cells, whereas in serum, these genes are much more homogeneously expressed. And you can kind of appreciate that visually, and we can quantify it in different ways and so on. And this sort of puzzled us because both the cells are proliferating both cultures, so why is there this difference? And then we realized after a while that they, in this passage of the ES cells, the 2I cells are cycling half as fast as the serum cells. So the serum cells are cycling really quickly, and we think that many of the genes just don't have the time to go up and down during the cell cycle. These cells are cycling so quickly that the cells are the genes from G1, S, G2M, and so on are just uniformly highly expressed. Whereas in the serum condition, where they're cycling more slowly, the cells kind of go through the cell cycle stages, and each module of genes for each stage has time to stabilize and be high in that stage and then lower in the other phases of the cell cycle. And that's why you get these differences in noise. It's a bit counterintuitive, but that's basically the explanation that we came up with, and that we actually validated also in other populations of cells where we also know their relative cell cycle speeds. So this is actually a pretty cool principle that we can extract from this, is that you basically have a kind of internal stopwatch of cell proliferation speed from your single cell RNA sequencing data. And you can estimate relative speeds through these, through the noise in the expression of the cell cycle genes. So obviously if the cells are quiescent, then these genes simply won't be expressed. But if they're fast proliferating, then you will expect a sharp expression and a high expression. And Florian already spoke in a session, Florian Bruttner already spoke in a session yesterday, and what he went on to do was to then, with Antonio Schaldona and other postdocs, Kedar from my group and Valentina and so on, had a nice collaboration across our three groups. So Olli Stegle, and John Marioni and myself were all office neighbors in EBI for a few years, was to develop, to compare different statistical methods for assigning cell cycle stages from single-cell RNA-seq data. And this is available as Cyclone software on GitHub. And what Olli also mentioned, what Florian also mentioned was that you may want to assign cell cycle stages, and that's what we were interested in, but you may also want to get rid of the cell cycle, or actually other processes. You may want to get rid of technical noise, for instance, in this way. And what he developed was a latent variable model approach that you can train on a known set of genes and then get rid of that variable so that you can focus on other processes in your data more clearly. And this was actually a real eye-opener to us, because when you get rid of the cell cycle, the differentiation markers pop out much more clearly, even in fairly noisy data. And that was a pretty cool insight for me, how powerful this latent variable modeling can be. And we actually now use that also in other analyses that I'll come on to later. Okay, so that's that. The cell cycle is leading to variation 2i. Fine. But remember that the global variation is the same in serum, where this module of genes is very uniformly expressed. So where's the noise coming from in the serum cells? Well, if you look at the gene ontology categories up here, these red ones are all to do with organ development. Now, in the serum culture, there's obviously no organ development, but there is maintenance of pluripotency, and there are cells that are escaping the pluripotent state in the cell culture. There's a small population of cells that's known to actually be differentiating. So you have heterogeneity in the pluripotency versus differentiation state within one cell culture. And when Ola and Jason Tsang extracted the bunch of pluripotency markers, these are the genes in this up to here, and a bunch of differentiation markers, actin, keratin, and so on, these are the genes down here. Then what you can see is that in the 2i and alternative 2i conditions, all the cells are in the pluripotency ground state, and these markers are highly expressed. Whereas in the serum condition, we can see that there are clearly at least 3 different clusters. The cells that are in pluripotency ground state have no differentiation markers, nanog high cells, and so on. Then you have this intermediate group which has a little bit of expression of the differentiation markers. These are these orange cells. And then the red cells is a small population that's actually on the differentiation path. And using this cell cycle speed estimation, we can see that they're actually resting. They've basically escaped, and they're not cycling anymore. And we can validate this in various different ways. One thing that we did was to do a second batch of 400 cells of VS to NPC differentiation. And these are these light green, dark green, black cells. And you can see that the cells in the static serum culture are overlapping basically with that differentiation time course. And this is Alex Tuck and Kedar Natarajan who did this. And then what I won't go into is that we also did a validation of the gene-gene correlation network using CRISPR knockdown. We were able to show that genes that we extract from correlations within the single-cell data are indeed necessary for maintenance of pluripotency, because when we knock them down, you get exit from pluripotency. So I think this also illustrates a nice point, is that by having this wet lab that I've painstakingly built up over years, we're able to both generate the data, analyze it, and then do validations like the CRISPR knockdown, which is really satisfying actually. And to sort of summarize this part, what I showed you was that when we test drove this single cell approach on these 3 different culture conditions of mouse CS cells, and this was really, to me, quite a technical exercise, but it threw up some interesting biological insights, and it's inspired development of new computational methods as we had to tackle different problems going through the analysis, is that there's equal cell-to-cell variation globally between the serum and the 2i culture, between the different culture conditions, but that noise has different origins, because there's the cell cycle module that's varying in the 2i because they're cycling slowly, and the pluripotency module that's varying in serum. And along the way, Johan and the group, together with Ola and Valentin and the others, developed a database. And I think it's also an important point how to serve this single cell data and how to visualize it. And this is kind of our first sort of attempt at that, which we think is useful for exploring the data and different individual genes and so on, which the stem cell community always asks about. So I've mentioned Ola Kolodziej, who's now graduated and is heading off for a postdoc, and she was a real superstar who combined a high expertise in both wet and dry work, and she teamed up with Kim and John Marioni, and this was a wonderful collaboration with our statistics neighbors at EBI. I've mentioned Jason Sang and Pentao Liu, who are fantastic stem cell biologists, and also Alex Tucker, Natarajan, and Mark Buhler for the validation in yes to NPC differentiation, and Tommy Ilicic, who developed the cell-align pipeline for processing single-cell RNA-seq data, and the CellID quality control package, which is on GitHub. Okay, so I've shown you that we can detect cell cycle and pluripotency subpopulations in these mouse CS cells, and so the basically approach of single-cell RNA sequencing is useful and powerful, basically, in throwing up new insights into subpopulations and into genes in this particular biological question. Now, I said at the very beginning that T cells are really a passion of mine, and this is how I started my wet lab. Essentially, was in studying mouse T helper cells, and this was because a postdoc joined the group who had a background in immunology and was able to culture these cells, and so we were able to access them and study them. And so we've been using single-cell RNA sequencing to study how T cells basically respond to an immune challenge, and how they then proliferate and how from a set of precursor cells in different lineages. And what I should say is that in CD4 T cells, so these are T helper cells, they're also killer T cells, CD8 T cells, but particularly in CD4 T cells, there's a lot of plasticity that I mentioned. This is what really got me into the need for single-cell sequencing kind of very early, before there was really high-throughput single-cell RNA sequencing. So Azim Sourani was the first person who used RNA sequencing on individual cells, and he started with about 4 cells. And it's the high-throughput mode that's made all this possible, and that's posed so many challenges for us computationally. So in this particular context, you don't need to pay much attention to this, but just to say that the precursor naive T cell that we kind of have in our spleen or in our blood and so on, when it's stimulated by antigens, so by a challenge like an intracellular or extracellular pathogen, or in an allergy or autoimmune disease, or by a neoantigen in cancer, then it will go down a variety of different lineages, effector cell lineages and also regulatory T cell lineages. And these can be Th1 and Th2 subtypes that I've mentioned, and also T follicular helper cells that are telling B cells to start making antibodies. And the particular infection that I'm going to talk about here, and this is again unpublished work that I'm sharing with you, is a malaria infection in mouse, and that elicits a Th1 and TFH response. So this precursor cell, when it sees the infected red blood cells that have the malaria parasite inside, it mounts both a Th1 differentiation and also B cell help through T follicular helper cells. And this is known, and we can recapitulate that with flow cytometry for particular markers for Th1 and TFH cells, and these are days post-infection. So the questions that are really important immunologically here, but also fundamentally in terms of understanding cellular decision-making and gene regulation in the context of a differentiation process, is how the Th1 and TFH subsets arise in vivo. So how exactly does a Th1 cell get here from its naive precursor? How does the TFH cell get to its end state from the precursor? This is something that's hard to resolve with bulk methods or with FACS and conventional methods that focus on individual protein markers. And then secondly, what signals and factors regulate these transitions through the intermediate states? So the experimental setup is that we have a donor mouse with a TCR transgenic engineered to be, where the two chains, the alpha and beta chain of the T cell receptor, are engineered to be specific for the malaria parasite, for particular antigens. So we know that those pink cells are responding to the malaria. They're not because our mouse house is dirty or something like that. And then there's a recipient mouse that gets these cells and that is then infected with the malaria parasite. And we profiled several days, up to day 7, which is sort of where the infection is starting to go down. The peak is sort of around here, and then also uninfected mice as controls. And we also profiled several types of cells in parallel, and I'll come back to that at the end, which made really interesting analysis of how the cells are talking to each other across time, basically. So just to start off with, if we do a simple-minded principal component analysis of the transcriptomes of the cells at day 7, then we can see that the principal component 1 roughly corresponds to differentiation. The TH1 genes are on the left-hand side, the TFH known marker genes on the right-hand side. PC2 roughly corresponds to proliferation and separates small from large cells, so roughly cell size, which we estimate through the ratio of the mouse, this is mouse obviously, transcripts to the spike-ins to get an estimate of transcriptomic size. If we look at, and we can kind of do a sanity check by comparing known TH1 and TFH markers and sort of saying what these cells are. Now what we want to do is to basically collapse the cells from all the different days, sort of I think 400 cells or so, into a single space and understand their development essentially. And to do this, yes, we could use PCA, but it turns out actually a much nicer way of, and much more powerful way of projecting the entire transcriptomes of these 450 cells into two-dimensional space is this Bayesian Gaussian process latent variable model, which Valentin Svensson implemented. And what this allowed him to do was to use all the 15,000 genes that are detected in these cells and describe each cell in terms of the two latent variables, which again, like the PCA that I showed you, roughly correspond to proliferation down here and T-cell activation going up and down on the y-axis. And what popped out, and you can see the different days, the cells from the different days are in different colors, and there's roughly a progression that's sort of circular here, and you can see that Valentin kind of traced this progression using a single latent variable to extract pseudotime. That roughly corresponds to the chronological time, but not perfectly. So the cells are, yes, they're all kind of progressing, but they're progressing at slightly different rates, so that some cells from sort of day 2 are intermingling with cells from day 0 and so on, because they're getting activated at different speeds. Then what I mentioned to you was we have a single sort of pseudotime here, but as you know, we've got at least TH1 and TFH cells, if not more, states in our final sample at day 7. We must have that. So we know that there is a bifurcation at some point in this developmental process. So how can we extract that? What Valentin developed was a really beautiful way of describing that using overlapping mixtures of Gaussian processes. And the optimization of extracting these processes is shown here, and this is a slowed down version of the optimization, so it's actually faster when you run it on your Mac. And you can see that there are 2 processes that pop out as optimal. Not 1, not 3, but basically 2 come out here. And we can describe the trajectory then in terms of these 2 Gaussian processes, which I think is really beautifully elegant because it gives us a handle on the function of this bifurcation process. And I should just spend a little bit of time saying that this is a machine learning method that was developed by Neil Lawrence in Sheffield and colleagues about 4 years ago to model missile tracking. It was applied to missile tracking. And so here, for the single cell tracking, we've got a slightly more sort of peaceful application of this process, of this machine learning method. It works also for trifurcations, for different complex branching patterns and so on. And applied it to simulated data and also lots of different published data sets. But I won't go into this in any more detail because I want to push on with the biology that we can extract here, and that is that these endpoints at day 7 of the two Gaussian processes correspond indeed, so the cells that we can statistically robustly assign to one process versus the other correspond to the TH1 cells in red and the TFH in blue. And the TFH seem to change less relative to the naive cells than the red TH1 cells. We can dig in more into the what's precisely happening during this T-cell activation process that goes from a quiescent precursor to a cycling intermediate state by looking at the number of expressed genes per cell, and this is the pseudotime on the x-axis and the number of expressed genes on the y-axis. And you can see there's a peak around day 3 or 4 where we have 5,000 genes expressed, whereas we start off with 2,000 and end up with also many fewer per cell. So the quiescent precursor cells and also the cells that are terminally differentiated and slowing down have fewer expressed genes per cell than the cells that are in that burst of proliferation in the middle. That also corresponds by FACS, or that also corresponds to the expression of Ki67, which is a proliferation marker. And we can validate this with the Cyclone software that I mentioned, and we can also validate it by flow cytometry for cyclins and so on experimentally. So that, I just want to summarize here and say that we can computationally, using the Gaussian process latent variable model for getting the main latent variables in the pseudotime and the overlapping mixtures of Gaussian processes, we can see that the fate bifurcation is happening around day 4 and then assign the cells statistically to the one Gaussian process and the other. And then really in the last couple of minutes, I just want to say very quickly that we can also use this kind of framework to dig into which signals, which external signals, and which intrinsic factors inside the cells are regulating the transitions through these intermediate states. So we can extract the genes that are most strongly associated with the TH1 process or the TFH process, and basically we get gene lists that are transcription factors, cell surface receptors, and other factors that are TH1 associated and TFH associated respectively. And so very interestingly here, ID2 is associated with TH1 and TCF7 with TFH, and indeed there's been a publication that validates that these two novel transcription factors that are associated with, there's a publication that's validated the TCF7 association with TFH cells in eLife recently in a different viral infection model. So that's a nice validation of that. And you can see that we can also use the Gaussian processes to trace the development of gene expression associated with the TFH process and the TH1 process for these two transcription factors, TCF7 and ID2. And the bifurcation point is roughly, we can I won't go into that, but we can kind of determine that as being here. And another thing that we can extract from this data is something that's really, really fascinating to me, which is the lineage of these cells, because we have a kind of intrinsic barcode that tells us which cells are sisters, cousins, second cousins, and so on, as the cells are proliferating, which are the endogenous T-cell receptor chains. So I mentioned this as a transgenic mouse with the alpha and beta T-cell receptor. There's also low expression of the endogenous T-cell receptors. And basically, as you're probably aware, in T cells and B cells, we have somatic recombination that takes place in the thymus. And so the precursor naive cells all have different barcodes of this CDR3 region in the endogenous alpha and beta chains. And so the cells that derive from the same naive precursor cell will have the same alpha-beta combination. And a few months ago, Mike Stubbington and my group published a paper with a pipeline for reconstructing the CDR3 region, for reconstructing the full-length transcript from SmartSeq data for the alpha and beta T cells. We call this Tracer. It's available on GitHub. And we apply this software to our malaria-derived CD4 T cells. And what we see, which is super fascinating, is that siblings, so basically cells that have derived from the exact same precursor, are present in the two different lineages. And you may think, well, that's kind of the default. Why shouldn't that happen? Some turn left, some turn right, kind of stochastically. But actually, it sort of goes against the dogma of what's expected because it's thought that the strength of signaling kind of biases the differentiation in one direction or the other. And Federico Sallusto did show in a paper in Science last year that this is possible in vitro, and here we have it in real life in a mouse going through a malaria infection. So just to summarize about what we've learned about the signals and factors that regulate these transitions, we can pull out statistically by association with the two processes, ID2 transcription factor and many other genes highly associated with the Th1 process and TCF7, and a smaller number of other genes with the TFH. We've shown that there's a highly proliferative precursor state, and that the same clone can give rise to both states in parallel. And there are, these are the intrinsic factors that we can extract statistically. Now, of course, these T cells are in the spleen here. They're seeing the red blood cells in the mouse spleen, and they're also talking to many other cells. And what are the signals that they get that then kick off the whole process and tell them to differentiate in different directions? Well, we profiled monocytes and also dendritic cells, and the monocyte data really gave us clues about how these cells can interact with each other, because we saw monocytes at day 3. There's a subpopulation that expresses CXCL9, a chemokine for which there's a receptor on the activated T cell, and that correlates basically with the Th1 fate. And so this gives us a clue that there are extrinsic signals from other cells that are telling, and we validated that, by the way, with a depletion experiment with a diphtheria toxin mouse experiment. Anyway, so we validate, so we can infer basically that inflammatory monocytes are signaling to the naive cells to go down this route. We know that B cells are interacting also with the T cells to bias them towards this route, and so. This decision-making process is really a combination of these landmarks from extrinsic cells, these cues from extrinsic cells, as well as the intrinsic factors that we can calculate statistically from the single-cell data. And I just want to say that none of this would have been possible without these very powerful machine learning methods that Valentin implemented, the BGP-LVM and for the pseudotime, and the OMGP for the calculating the cellular trajectories and these regulatory factors. And so again, this was a beautiful collaboration between computational experimental people in my group. So Tapio is a Finnish postdoc who did all the wet work, Valentin, and did a lot of the dry work, and Mike Stubbington for tracer. We collaborated with a malaria group in Sanger, Oliver Bilko, and Rudy Montandon. Ollie, whom I've mentioned before, was fantastic for support in machine learning, and also with input from Neil in Sheffield. And then Ash Hawke and Bill Heath and Kylie James, also a fantastic postdoc in Australia, for the TCR transgenic mouse. And so just to summarize, I've talked about assessment of different protocols using spike-ins to compare their sensitivity and specificity. And I've talked about how we can analyze the cell cycle in different subpopulations in ES cells and serve that data with the Espresso database and the distance to the median method for calculating expression level regress noise. And then for the T-cell decision-making, I mentioned the tracer method very quickly and the overlapping mixtures of Gaussian processes. And I'll just finish with this slide, and thank you for your attention, and I'll take questions.